First of all, a light introduction to the EEG. It's record the activity of the cortex, the electrical activity of the cortex. So it's an indirect measure of cortical activity. And as following the example of Alex or the example of our colleagues, the previous speaker, the EEG, maybe our objective is try to characterize the cortical activity in or some particular function, but the EEG involve the sound, in this case, of a lot of people. As the previous speaker says, as local field potential is a part of a stadium, so the EEG is the whole stadium. So in this case, I will focus on the event-related potential. That is very, sorry. There are specific periods of the EEG that are to an event. For example, in this case, we have the continuous recording of the EEG. There are a program that shows you some events. For example, in this case, the letter X and the letter O. So we can identify some periods, time windows, where we can extract the information that relates with this external stimulus. This is another example. In this case, we record the continuous EEG, and this is the task. And you must to press the mouse button if the circle is red, and you must to inhibit when the circle is blue. So we need the continuous EEG, and also we need the markers. That is this kind of marker. Then after this, it is necessary to perform the trial segmentation in order to obtain the ERP. There are two main strategies for analyzing ERP data. One of them, for example, for the continuous EEG data, we are segmented in trials, as we said. These trials are synchronized with the stimulus. It is commonly known as stimulus onset. Okay, so these different trials have different the information of different channels on their skull, and we can reorder this information. And here we have only one electrode, and we can order all the trials related to the trials of the experiment. So we have different matrices for each electrode. So in this case, from this point, we can perform the average evoked potential, that is the trial averaging of these trials, in order to obtain some components, or we can work with the single trial analysis. So each one of these trials, we perform a time frequency analysis on one of its trials, then we can obtain this time frequency maps, and then we can extract some filters and perform some statistics. Or I will enter to the slide both approaches. The first one, as I said, the main point is the averaging of the trials, but on a single trial, it's not possible to see a clear behavior. So it's necessary to use more than one trial, of course. In this case, we have some representation of the average of this is only one trial, the average of 10 trials. 10 trials, 20 trials, 30, 40, and 50 trials. So we can see that as we increase the number of trials that we are averaging, the signal looks like the final one. So one of the open questions is how many trials are needed in order to obtain reliable event-related potential. This is the same idea. In this case, we have a non-target signal, and the blue one is the target signal. As we increase the number of trials in average, the signal is more marked. The typical visual of what is that you have some distractors or standard images, and there are the target one that is the image of interest. What are the pros or the cons of this kind of analysis? Of course, the pro, one of the better things is that we reduce the signal to noise ratio by this averaging process. So and you can see there. And also we can obtain a signal with a good quality, and you can try to characterize some different dynamical behaviors based on this analysis. About the cons, if you use a lot of trials, you can contribute to be fatty. So for this reason, it's very important to analyze the minimum amount of trials in order to achieve a good quality of the signal, but try to minimize the fatigue. And also, as the experiment was done, it increased the negative of impact on the task performance. How can we perform? We represent this kind of average potentials. You have a battlefield plot in where we have representation of the amplitude of this potential along the time for all the channels. Here there are all the channels. So we can select only one electrode or even a region of interest. So averaging more than one electrode, and you can obtain a single plot, or this is the amplitude, this is the time. Or we can select a time window of interest. For example, we can select a time window from 400 milliseconds to 700 milliseconds, average the signal on these time windows, and then we obtain a topographical plot. There are the three main ways to represent the average evoked potential. The nomenclature, I will pass a little bit quickly. Basically, it depends on the polarity of these potentials. P, it means positive peak, and negative peaks, and the number represents more or less the time in milliseconds. So the P300 is a positive peak, more or less, that occurs more or less in the 300 milliseconds, or maybe the N100. What kind of potentials used to obtain? The first one are the exogenous potentials that are related to the sensory processing of the task, of the cognitive task. This there are the early waves, normally until 100 milliseconds, related with the stimulus onset, and this is, for example, typical the N100, and it is mandatory. If you see a target image or listen a target sound, N100 appears if you have attention. But there are another kind of potential, this endogenous, and this is the late waves, and in this case, it evaluates the subject response to the stimulus. So in this case, the subject is performing a task, for example, responding to pressing a button or something like that. So in this case, this is involved in processing information. Between in the analysis of average evoked potentials, we are focused now on motor-related event-related potentials or motor-related cortical potential. In this case, sensory ERPs are aligned to the stimulus onset, the moment where the image appears or the sound appears, but in the motor ERPs, they are aligned to the response or even to the movement onset. Then Monica will explain the difference between the response or the movement. So in this figure, there are the same data. It's a motor task. It's a motor task, and this is if you align the trials related to the stimulus onset. In this case, we have the average of the response time, and this is the butterfly plot that we obtain. But if you align there when the button is pressed, the potentials are different. So there are different. It's important to choose the correct alignment. Okay, depending of the task or depending of the purpose of our research, you can use a stimulus onset, response onset, or even movement onset. In this case, the motor-related cortical potentials is a type of ERPs related with the movements. And there are two kinds of categories: self-initiated or stimulus related motor movements. In the stimulus-induced movements, evidently, there are an stimulus that mark when you are to move a piece or to perform some movement. In this case, there are a potential that is called contingent negative variation, the CNU, that appears 100 milliseconds post stimulus, but as we align to the response, it appears just before or at the moment of the person is executing this action. Okay, in this case, this is aligned, but with the moment onset. Okay, and this is the signal. And we can characterize as well the self-initiated movement. In this case, we don't have any stimulus that mark when the user must to move or to perform a movement. So in this case, this of course is aligned to the movement or the detection of the movement, and this is a potential that is it's related with the preparation, with the intention for this we have said reflex conscious intention of decision-making processes. So it's the intention of making some movement. Also, we can speak about the sensorimotor rules that are related to the BCI, motor execution or motor imagery. In this case, we have two types or sensorimotor rules that are mainly known. The ERD is the synchronization of the power of in some frequencies, and this is the synchronization of the power of some frequencies. And it's very important to specify the frequency band where you are analyzing. The Bose studied frequency bands at the view at the beta bands. In this case, this is a result from a partner in Barcelona. In this case, we are studying the synchronization and synchronization in the mu band and also in the beta band. So we can see this desynchronization and then the synchronization in the signal. Also, we can analyze it by using a single trial approach. In this case, we can obtain a time frequency representation, so we can observe the variation of the spectral power along the time in the different frequencies. As I mentioned, the sensorimotor rings are very used in motor imagery approaches. And as I mentioned before as well, the number of repetitions is very important. So we have published a study, a paper related with assessing how many repetitions, how the repetitions impact the motor execution and motor imagery on a public that analysis. And is here the results of this is the number of trials. Increasing the number of trials, we have you can observe that we obtain less variability, okay, and also this is the topographic map. Increasing the number of trials, we can characterize better the areas related with this model. But it's not necessary to go to the top, to the maximum number of trials, because maybe with a number of trials between in the middle, we can characterize and localize well this kind of potential. Now it's time to go so to single trial analysis. In this case, why is a good option to analyze trial data? Because there are some factors like fatigue, attention, emotions. If you are performing the average, you lose this information. Also, to evaluate the dynamic process of the brain, to analyze rare events, and it enhances the signal statistical power by introducing some features related with the magnetic frequency. So, as a summary, single trial is very suitable when when we have individual variability. We are interested on evaluate temporal dynamics and also if the rare events are of interest. What is more or less the general approach to analyze single trial ERPs? Of course, the first step is to extract the trials. We can apply a time frequency analysis, for example, short time Fourier transform, Wavelet, Stockwell, or even another kind of time frequency representations. It's very important to take into account the Heisenberg or uncertainty principle. So, for example, with the Wavelet transform, we must use the cone of influence, okay, because we are, as we are filtering the data, it's important to take into account the window of this filter, because if not, you can obtain withdraw results. Then we can perform the feature extraction. That is, you can start a lot of features. We can perform or not this feature averaging. Of course, then the last part is to apply some statistical analysis on them. One of them, this is some representation of frequency plots for ERP data and the topographical ones. But one of the main parts of the ERP, the single trial analysis, is that we can perform some phase-based measures. When you average the trials, you maintain the amplitude of the measures of the trials, but you lose all the information of the phase. Okay, and there are some analyses in the field of psychiatry or neurology that say that there are very important information in the phase. So in this case, it is recommended to use the single trial analysis. That is that some challenge when you are working with single trials, for example, the quality of the signal, or also the filter, because, for example, if you use an EER filter, you are modifying the phase. So it is not convenient in this case, some different consideration. And also the analysis of single trial data open you the opportunity then to perform some connectivity analysis and also graph analysis on the image. So I have explained the average evoked potentials and single trial analysis, but what's happened with the BCI methodology? BCI approach are between two of them. It does not use only average and it does not use only single trial analysis. So from BCI data, you can obtain some stimulus log or response log potentials. In this case, we can use the amplitude, the latency, and performance of localization. And so we can obtain these motor potentials. But we can work also applying this, some filters, applying the common spatial pattern in order to obtain some features related with the single trial analysis. So in this case, a typical analysis with the average potential is the speller, the T300 speller, and a typical analysis using a mix of these both technologies is the motor imaging, for example. And also the BCI approach introduce another dimension that is the calibration of the training of the common spatial pattern features, and also the extraction of the features from the classifier. And then we will apply these configurations on real-time processing. So it includes more complexity on the data. About the common spatial patterns, only one slide, and it tries there are it's a kind of filters that try to differentiate between these classes based on the covariance. So it tries to differentiate these places, and it's a good tool in order to separate the signal from the noise, in order to clean the data and to give cleaner information to the classificators. Now it's time. I don't know how about the time. It's time to present two research samples of our group. One of them is related with the stroke rehabilitation. The other one is a particularization because it's working with the Rett syndrome, that is a rare disease, and there are some particularities. The first one is the stroke. Well, it's as this morning Hattie said, it's a pervasive disability that cause a great impact in the quality of life and the independence of the patients. So the motivation of this study is to perform a cognitive rehabilitation in order to try to improve the quality of life of these patients. Regarding the phases of the stroke, the acute phase and subacute phase is the best time to recovery, and this time where this project focus on. In this case, we have 4 sessions of cognitive therapy with EEG, and also there are a neurocognitive evaluation by experts only in the first one and in the fourth one. And each one of the sessions is more or less 1 per week. 1 per month. We have the system that we use is the Autobiolab, and we perform it in the Goodman Institute. We have developed a system, an event system where there are different kinds of tasks one related with short memory, this is the inhibition, this is related with the lexical decision task, it is related with the vocabulary, that is a very important thing, the mental rotation in order to visual spatial tasks. And in this case, I will present the preliminary results for this task that there are, it appears some words. Some of them are real words, and some of them there are no real words, but it uses some common phoneme. I don't know how to say in English, but some common structures in the language. For example, it can be an no word, and this is a word. And the patient must to try to respond if it is word or it is not word. Regarding the percentage of correct answer and the response time, it is the difference between seasons. This is the first section today. The second one, and also this is the response time. Here we can see some different potentials that we have evaluated, and we are comparing them between word and pseudo word in the first session, in the first register, in the second one, and also a comparison with the control. The same is for different potentials, and we can see that there are a recovery of some functions when the sessions is another potential. This is the Rett syndrome results that I will pass a little bit quickly. In this case, this rare disease has the particularity of they don't have the ability to communicate. So in this case, we have a link some software for stimulating this kind of behavior diseases, and also we will use the eye gaze in order to be sure that the patients are following the task. When we are performing this approach, we are registering the EEG data. This is an example of the approach, and we have some difference: an active task and also a passive task. The active task, for example, is there. It appears some image, and these girls following by the eye gaze, they can to move along the task. And passive, for example, in this case, the therapeutic therapists say, for example, ball, and the girls must to fix their eyes in the ball. So there are two kind of treatment, and also there are different. This longitudinal. The treatment is longitudinal. This is the publication that we have, and we can see that this is the first, the basal, the first register, the second, the third, and the fourth, and you can see that a reduction in the low frequencies spectrum, power spectrum, and improvement in the high frequency. This is the same for, this is separated because of the amplitude, okay? And the reduction of lower frequencies and the increase of higher frequencies is related with an improvement in the parameters that are measured in this study. Sorry, I don't know how. Okay, and this is the more or less the same, but in this case, representing the localization of this difference. In this case, the passive task does not stimulate a lot, but the active task. In the active task, you have some difference, and the main difference that we obtain are in the resting state. So the conclusions were that by stimulating these girls during several days, we can obtain that their basal activity, the resting state activity, are there are some changes in the resting state activity. So it's time to my partner, Monica.
So we wanted to present some initial results of our exploratory research project and also the experimental part that we are performing at Goodman Institute. So the idea of this analysis is to try to study the mechanisms associated to stroke rehabilitation. So we are recording signals and EMG signals in patients after the acute case during the rehabilitation at Goodman. That takes more or less 3 months. Okay. We are aiming for 10 control subjects and 10 stroke patients with one evaluation at baseline and after 4 weeks and 8 weeks of treatment. And we evaluate force, motor execution, imagery, and in isometric conditions, and then motor control in dynamic conditions. So this is the first part of the experiment, which is related to motor execution and motor imagery. In this case, it's isometric conditions. So we are using mechanical brains. Okay, thank you. I can put, okay, like this. So the subject is presented by the stimulus on the screen, and then he has to perform an isometric force in the 50%, but 50% of muscle maximum voluntary contractions. And since we are analyzing potentials, we do this for 16 trials divided in 2 hours. Okay. They have 6 minutes of 6 seconds of contraction and then followed by seconds of rest. And we tried to use at least 6 hours because we wanted to calculate the potential. But that is actually a little long for these patients. And then they do this motor execution, and then they start to do the same but with okay, and then they also have an endurance test to analyze fatigue in the The second part of the experiment is the dynamic contractions. So we designed this three-dimensional plane where they have guides to move. In this case, this patient is able to more or less control the movement. There are some that not. This is the first session of the experimental protocol, but we have seen that for the third session, we have recovered alert. So it has some guidance in here. It's guided, but it's not very constrained. So we were aiming at trying to study this in normal conditions in something related to daily activity. Okay, and then it has to follow these lines on the plane, and we are measuring signals in the EEG. We are measuring also all quite a set of superficial muscles for the control of the shoulder, which is very interesting for the physicians, because this is one that is most here, that's the stroke. So we're measuring the deltoids, the pectoralis major, the biceps, the triceps, and the trapezius. So we use a lot of channels, and this is one of the how do you say, the the biggest challenges in the protocol, because it's 400 channels we are measuring, and the preparation is very intense. So actually, the preparation takes more or less the half of the time of the protocol. And we have here some preliminary results. These are from more or less a month ago or more. So these are only like 6 subjects, it's patients in each group. So it's not a statistically, it has not a statistical power. But as I said, we are in for patients, okay? And the first was to analyze the motor-related cortical potential. And the first thing was to try to align the potentials with respect to some signal. So we were using this here. The red line is the stimulus presented in the screen when the patient is supposed to start to contract. But we have seen that using this for the alignment didn't bring a very clear potential. This is the potential that we obtained for the 6 subjects. And most of all, this negative peak is not very big. Okay, so we decided to try with other signals. We used the torque signals, but it didn't work very well. So I'm not presenting here the results. So at the end, we decided to work with the envelope of the EMG signal. So we are aligning in here. We detect the onset of the contraction, and we use this onset for aligning the different tracks. And if we do so, we obtain this more nice shape of the potential that we were aiming for. And this is actually in CC in this. So these are some preliminary results, like I said. So these are the motor-related cortical potentials for the control subjects. The potential is very nice. Actually, the potential is a solid line, and the shadows is the variability related to different subjects. And we can see that the potential is very localized in this area, where it's supposed to be localized. And then we do the same in the patients. Actually, we don't observe a lot of difference. Mainly, the biggest difference is the amplitude of this peak, especially in the first trimester after the acute phase, which is very high in the patients. But as the potential is less, it can maybe is improved. But the most important results up to now is in the event-related desynchronization, either in the beta or in the mu band. So this is the potential in the control subjects again, and the localization is mostly in the contralateral side. I also forgot to say that we have patients that have a dysfunction of the left or the right hand, this is the control. So actually, this is all directed to the right hand. Okay. And we can see that this peak is mainly reflected in this area, as was expected. But then for patients, especially in session 1, these potentials are very spread all around the skull. But after seizure number 3, we can see a similarity between these two. Okay? And just with this number of patients, that is more or less 6, we can get almost significance in these differences. But we are continuing working on that. And the same happens in the mu band, which is actually better in this band than the band. Again, so and the amplitudes are more or less similar to the control subject. And afterwards, we also have the synchronization. So what happens after the termination of the contraction or during the relaxation? And in this case, the results are not very good, but I also have to say that this was aligned again with the movement of And perhaps some of the patients continued for longer time, so maybe that is one of the problems, and this needs to be corrected. And the same happens in the new one. Then we tried the same during neuroimaging, the different lines correspond to different patients. But in this case, none of the potentials, the motor-related cortical potentials or the event-related desynchronizations are clear. One of the problems is that we don't have any means of aligning the triangles. So we have to find out the techniques. Actually, what we are using for the alignment is to obtain the average and delay after the stimulus for a subject. But these potentials are not yet very good, so also we will try other different methods that are not related to potentials, to evoked potentials. Okay, and I think this is it. Okay.